Yeah, so maybe I'll just go ahead and introduce Professor Yiliang Ding. We are pleased to welcome Professor Yiliang Ding from the John Innes Center, United Kingdom. And to just briefly introduce her work, so her group is particularly working on studying RNA folding functions, especially the regulatory role of RNA in living cells, and they're also interested in RNA-based therapeutics, I think particularly in crop improvement. And she happens to be the first UK plant scientist to receive the prestigious Blavatnik Award last year for the young scientist. So, first of all, congratulations, Professor Ding. And we are looking forward to your talk. If you're ready, please go ahead and start presenting.
Yeah. Yeah, thank you very much for the invitation. Actually, I know the CASP for quite a long time, but I didn't really know there's actually seminar series on that. So it's very honored to actually give share our study here, and hopefully it's not really towards a 3D structure prediction, but we actually more focus on the functional kind of RNA structure element. But I think it would be really good combination in the future. We actually how to have accurate prediction of a structure, then further on, understanding the functional role. So basically, I'm going to talk about several dry bench and also wet bench method we developed in the past couple of years. And just to give you an idea how we actually doing all these functional RNA structure kind of study. So basically, I don't need to introduce too many things about, we actually want to understanding how RNA fold. So almost all the textbooks do draw RNA as a single line, but we know it's not the case. So we basically have a stem loop, also, more complicated 3D structure. For years, I think the most limitation part on understanding all these kind of a structure and their function is mainly just because it has been doing a lot of structure study in vitro, but not in vivo. So it's really about a global effort pretty much 10, 11 years ago. So I think our group and also Suvoroskin and Danny Incado, Robert Spetto, also Yue, crossing the whole world, there's a lot of young scientists that we actually come up with this kind of concept called in vivo RNA structure profiling. So basically concept are very simple. So we're using both dimethyl sulfate and a shape. So dimethyl sulfate again can actually target to the single-stranded A and C nucleotide at Watson Crick phase. So then the method group will attach to the Watson Crick phase. So you can capture by reverse transcription stalling, or some recently we actually can do through mutation profile. And through a lot of kind of development from Kevin Weeks' lab, so if you're doing DMS reaction at pretty much pH 7, 8 kind of condition, you can actually now probe U and G as well at Watson Crick phase. So the other chemical is from Howard and Kevin's. So it's a shape chemical. Mainly, we actually work in plant mainly on the Howard's NAI. So this chemical is actually isolate 2-palm hydroxyl group when it's single-stranded. So similarly, you can capture by reverse transcription stalling or the mutation profile. Through deep sequencing method on the Illumina in the past, 10 years, a lot of application has been done in. So basically, you can using adaptive ligation towards to capture the single-stranded DNA on this modification position. So you will be able to count how many reads. If you have a lot of reads stalling at a specific nucleotide, then you can generate something called a chemical reactivity. So if you have a higher chemical reactivity, that means this nucleotide is tended to be modified by chemical, which indicate this nucleotide tended to be more single-stranded. Oppositely, if you have a low chemical reactivity, have less reads storing at the nucleotide, a specific nucleotide, then you can actually get something which is a low chemical reactivity, indicate the nucleotide tended to be more double-stranded. So similar with the mutation profile, you have a more mutation, likely to be more single-stranded. If you have less mutation, then you have likelihood to be more double-stranded. So when I started my lab pretty much 11 years ago, we really actually looking back towards textbook. So through the transcription towards processing and the translation RNA degradation in plant, we also have these microRNA mediate RNA degradation pathway. So basically, we want to see how much we can actually update this, textbook, which the RNA is not only like a single line any longer. So it was quite ambitious. So, but through years, we kind of get a little bit more kind of understanding. So my group are mainly working on very specific model plant species called Arabidopsis thaliana. So around the time, we're also working on different kind of plant species. So one point we're very interesting, I think through years, we kind of like the plant system is because plant normally, majority plant species habitat roughly 20-ish degree. So which means at that temperature, a lot of structure will actually thermodynamic fold quite stable compared to 37-degree or more high temperature. So that's why currently it's starting to become more kind of a favorable kind of a system. We actually investigate the function of RNA structure. So I want to go through detail towards all these kind of study, towards update a little bit on the textbook. So we did a pre-mRNA structure. So we found a certain structure feature associated with splicing, polylation. Also, we find a structure motif which are associated with the translation, RNA degradation, also microRNA mediated cleavage. I think even in 2019, we actually published RNA-driven phase separation based on these specific motif called G-quadruplex. So today I'm gonna focus more on the G4 G-quadruplex. It's more 3D structure, so I thought I give a little bit more kind of associated with the similar theories. So G-quadruplex is a very specific structure motif. For years, people notice it can form into these kind of a beautiful tertiary structure where you have a G-rich sequence motif. So what it folded into is you have a guanine, you're using both Watson-Crick phase and a Hugenstein phase. So then you can form these Watson-Crick-Hugenstein base pair, two bond. Then you have these kind of 4G folding together as a plane structure. So if you have one layer of that or two layers or more than two layers, 3 layers, 4 layers, you can actually form this kind of a beautiful tertiary structure. So if you're looking for the sequence, it's actually 4 times G2 repeat. So you have a GG, then loop sequence, GG, loop sequence, then GG, loop sequence, GG. So if you can have a 3-layer, humans, you have a more like a G4, G5. So if you search this motif sequence, it's everywhere in the transcriptome, also genome. So for years, people also want to know whether or not it's truly folded inside a living system or not. So this structure motif really specifically fold much more stronger when you have in the condition of potassium. If you have lithium, because it's too small, it couldn't hold in the middle, so it will destabilize the structure. So why we actually started interested in this kind of a G-quadruplex motif is really back to 2016 time. We actually noticed this shape chemical, which I talk about acylate 2'-hydroxyl group, is actually have a very specific modification pattern on the G-quadruplex. So when G-quadruplex is folded in the potassium condition, the last G, the 2'-hydroxyl group of every last G of a G repeat, you can actually see it's actually got a specific modification with this NAI shape chemical. So because of this topology, it's opened this 2'-hydroxyl group more exposed than the other Gs. So we can actually see this kind of a shape chemical modify this last G more preferentially. So then through reverse transcription stalling, when you capture the stalling signature on the reads after sequencing, you will see these ununiform distribution, very uneven distribution here. So then when we actually put the same sequence into lithium condition, which you destabilize this G4, you can see you lost this preferential modification of every last G of every G repeat. So you got a more even distribution. So then we thought, okay, whether or not we can set up a, in vitro folded standard for the whole transcriptome and an in vitro unfolded standard for the whole transcriptome, then compared to the in vivo, right? So then we actually did a in vitro library with under condition, in the lithium condition, also the potassium condition. So this is one of the G4 we validate by physics analysis. So we know it's folded. So you can see under lithium condition, you have a more even distribution. Then in the potassium condition, you have a more ununiform, like a kind of a pattern. So then we thought, okay, maybe we can now using this as a standard to actually assess how the in vivo probing looks like. So if it's similar to the in vitro folded, then we call folding score as 1, like it's likely to fold in vivo. Then you actually align with these unfolded status, then our folding score as 0, then that means it's likely not folded in vivo. So again, towards the example I just show you, see this the same G-quadruplex, you can see it's actually more similar to the potassium. That means this G4 is likely to actually form. So how about a genome-wide? So we can calculate every single G-quadruplex based on this kind of folding score. You can see most of Arabidopsis predicted G-quadruplex is more close to 1. It means it's more similar, kind of a 10 to fold most G4. If you actually see these high GC content kind of a genome, which is rice, for example, monocot plant, you can see the folding score is much more higher. So that means it's have a more G-quadruplex and the folding strength is much more stronger. So then we're wondering, because if anyone actually noticed in 2016, actually David Batel's group said using DMS-mediated method, they claim in the mouse and the yeast there's no G-quadruplex form inside the living cell. So we thought of why we can capture so many, plant in the plant we can capture G4, right? So then we did a geo analysis. So there's a lot of stress response, salt response, water depletion comes out. Interesting thing is you actually can find something in response to cold. So my postdoc said, is it because the plant, living roughly 20 degrees, so we can capture more G-quadruplex? So if we now drop the whole plant into the cold, what is going to happen? So then we put the plant into 2 hours at 4 degree. Again, we chemical probing with this shape and compare the shape NAI pattern, modification pattern towards the in vitro folded and the in vitro unfolded standard. Then we just see what happened genome-wide. So interestingly is at the 22 degree, some G4, G-quadruplex doesn't really not folded. It's very similar to the lithium condition, but when you actually have 2 hours in the 4 degree, you see it's very similar to the potassium folded status. So we know there's actually G4 can, G-quadruplex can actually induce the folding in response to cold. And if we actually take a look at genome-wide, you can see at 4 degree, you can see a shift of a folding score towards more stronger folded globally, crossing the whole transcriptome of Arabidopsis. So then we know G-quadruplex can globally actually induce the enhanced crossing the whole transcriptome in response to cold. At the very beginning, we think 4 degree, what is going to happen if you have a transcriptome-wide G4 form? So what are their function, right? Whether or not they are like a lot of people think G-quadruplex suppress translation, you will actually see the response in the code. But interestingly, when we did a translation, ribosome profile, also polysome profile, we didn't find any actually significant change on the translation efficiency. But instead of actually when we actually go for the RNA stability, what we find is when we're using transcription arrest assay at the 22 degree and 4 degree, we can actually find that there's actually quite a big change on these cold-induced G4. This is quite a complicated plot, but I just take home message is these cold-induced G-quadruplex located in the 3-UTR, these gene in the 22 degree is quite a fast degradation. But when you actually put into cold, it's folded as a G-quadruplex, you see it's getting very stable when it's going to the 4 degree. So then we're wondering whether or not, cold-induced G-quadruplex is try to stabilize RNA in response to stress. So then we actually did a reporter assay. So we put a reporter gene, and then after that, we put this 3-UTR contain this G4. code-induced G4 and also mutate a couple of G to A, abortion the capability to form G quadruplex. What you can find because it's only folded in the code. So when in the 22 degree, both wild type and mutant, you have a faster degradation. But when you actually in the code G4 forms, you have a more stable actually RNA going more stable. Then in the mutant, which abortion G4, you can see it has faster degradation. Okay, so as a plant biologist, we actually interesting to see whether or not it's really working inside the plant, right? So normally people will actually knock out the whole gene function. So we thought, okay, whether or not there's any function of this G4 for the gene function. So we take a plant, with the mutant plant, which don't have this gene, completely knock out. So no expression of this gene. So we complement this mutant with the whole gene contains 3-UTR with this code-induced G4. And we also complement the plant with the whole genes with the 3-UTR contain the mutated G4. Okay, we want to see what happened. At the 22 degree, you don't see different. 4 degree, when you have a Y-type plant, so normal plant, you will see when you have a cold 4 weeks treatment, you will have a shorter root. So it's a cold response of a plant. When you don't have this gene, completely you don't have this gene, you see much more longer root. It's like an insensitive to cold response. When you complement the whole gene with 3-UTR contain Y-type G quadruplex, code-induced G quadruplex, you can see it's actually rescue this kind of long root phenotype, so it can sense in the code. That's why the root are getting shorter. So when you complement the whole gene with the 3-UTR abortion the code-induced G quadruplex formation, you can see it's very similar to the knockout mutant because in this mutant, your RNA going through faster degradation. So that's why it's similar to the knockout, the gene phenotype. So you can see that we can just changing a little bit, non-coding region with the functional motif, you can actually abortion the code response. So then my postdoc wondering, if that's the case, we have these plant, 1,000 plant located to different location crossing the whole global. So whether or not all these plant transcriptome have some pattern of enrichment of G quadruplex, whether or not they select this kind of G quadruplex motif, right? So what we did is we predict 1,000 plant transcriptome, which we actually predict G quadruplex crossing the transcriptome, how much density of G quadruplex in their 3-UTR, 5-UTR, coding region, and associate their climate information. So basically, their annually mean temperature, their habitat temperature, which location their minimum temperature. We actually coordinate all the temperature and the precipitation, which is how wetness of the location, right? Very interestingly is we can find a significant correlation of G quadruplex density associated with the temperature, anti-correlated, which is very surprising to us. So, which means is, sorry. So basically, it means is a plant species, if they habitat in a very cooler area in the globally, in relatively cold environment, their whole transcriptome have more higher density of G quadruplex, particularly in 5-UTR and 3-UTR. So somehow we think, the plant habitat, because they have to adapt to different location, they might are using this strategy to actually enrich a certain motif, which allow them to actually facing stress. So it's really go back towards the time when we about to submit the work in 2021 in the RNA Society meeting. We actually presented just after our talk. Actually, Batel's previous postdoc, Junjie, now in Yale, and also Pavi's group in Harvard, they actually find very similar things happening in the human. So if you still remember, in the mouse and the yeast, you don't find the G quadruplex form. In the human cells, 37 degree, you also don't detect the G4 formation. However, if you put a human cell under stress, you can see the G quadruplex forming. Where it's induced mostly is actually through UTR. What these stress-induced G-quadruplex do, they actually stabilize RNA in response to stress. We kind of find this scenario back to back in plant system and also in the human system. So this is the very nice kind of a co-submission we actually work with. So we think RNA G-quadruplex is likely to as mostly like a universal kind of a stress sensor. So my group actually built up this G4 Atlas, which is for the whole community, so people can looking for experimental data on this kind of G4. So we recently working on another interesting tertiary structure motif. It's still on the DNA level, but we are working on towards RNA as well. So it's called IM-seeker predictor I motif, which is a C-rich motif. So I won't go in details on this part, but you can take a look. We develop also tools to predict this kind of C-rich I motif structure. So now I really want to go to another new technology which we developed pretty much several years ago, also quite a recent. So basically, if previously you see all the structure we study and a lot of a group publishes on the Illumina sequencing, right? So basically, for structure people, we always want to know is how much you can capture RNA structure dynamic. So this is one sequence in silico, how much possible conformation you can predict from Vienna fold. Okay, so if you're sampling out all the folding possibility, this is one sequence, how many potentially, structure conformation that we're folding into. So for a very long time, we were wondering how to obtain individual RNA conformation in the living condition. So we developed a new method which allow us to do that, but using a very interesting long non-coding RNA to demonstration this whole technology. So I just go over to this very famous plant long non-coding RNA called QL. So what QL do is during the plant vernalization, so you need to transition from the vegetative stage to the flowering stage. So what actually happening is during the winter, the plant will going through a very long cold period, which we call vernalization. So then cold will suppress a very important gene called FLC, and then you will have a flowering come out. In the FLC, how you actually suppress FLC is through its antisense non-coding RNA called QL. So QL will actually associate with FLC chromatin, then you silence the FLC expression. So for the time when I joined the John Innes Center 11 years ago, we actually thinking, okay, we should probing this QL because, the famous flowering lady, Yiliang Ding, is on site, so we should work on this QL. But if you're looking closely, this antisense non-coding RNA had pretty much 4 different isoforms. So you think about it, if we're using Illumina sequencing method to probe the QL, we don't know the structure coming from which isoform. So then we thought, okay, we just actually make a new method to specifically study this QL structure. So we generated some method called a single molecule-based RNA structure sequencing. So basically, again, it's chemical treated plant, all the single strand will be modified. You can capture all the 4 isoform. Then each isoform will have RNA population, which get a chemical modification. Using mutation method, you can get a single cDNA mutation. Then you can actually generate double-strand DNA. Then you can plug it into PacBio. Nowadays, we can do in the nanopore system. So you can get the reads out. So each hi-fi reads contain mutation profile. We can use in centrifold to a contrafold. We can actually generate an individual structure from that. So then basically, if you're sequencing 1 million reads, you end up a pretty much unique mutation profile. You pretty much generate a 1 million structure. Then you cluster with the structure similarity. So you can actually find the major conformation of each isoform. So this is one of the QL isoform, how much kind of a structure conformation we can find. We can cluster into, find like a 3 major cluster. So the question is really about, okay, this is a cold important, like a response code stress important kind of long-non-coded RNA. So if we put this whole RNA into code, what happened? So what we can find is actually two scenarios. One is the proportion of a certain conformation going down, certain proportion going up. You also find a very stable code response structure conformation. So based on this high resolution of a single molecule, measurement, you can get a very lot of detail towards, stability, also diversity of a conformation. So based on that, we took an area which we think this is a highly dynamic area, which we find quite an interesting area. We want to do some mutation. So we basically made a 4 mutation, make this whole RNA bigger. So the QL much more single-stranded. So then we also confirmed with this single molecule structure profile. So if you make this non-coding more accessible, so it can associate with FLC chromatin much more stronger. So when it's associated with FLC sense chromatin more stronger, you can suppress the sense gene much more stronger. Of course, you can get early flowering. So basically, that means if you actually find the right position of the functional region and tuning the structure well, so you can actually really have a strong genetic impact. So where we actually doing that, meanwhile, my lab accumulate a lot of data set. So you actually end up a lot of sequencing data set, and now we have a single molecule data set. So really about 5 years ago, I was starting worrying how possible we can best using all these, probing data, structured data. So one of the questions is how we can actually identify functional important structure motif, which actually identify crossing the whole transcriptome. So this is really go back towards a very famous method called AI-driven foundation model. So I think people now, everyone using that ChatGPT draft your email data analysis, also reading paper. So we were thinking whether or not we can actually produce similar things for the RNA sequence. So take everything simple. I think a lot of you actually already know how it works. So basically, traditional machine learning, deep learning, it's more like if you want a baby to learn Shakespeare's poems and want a baby to, write down a Shakespeare-style poems, it's very difficult, right? So, but if you actually train this baby with all the literature, grammar, English, classicals, so the baby become a PhD in literature. So now you feed in 1,000 Shakespeare poems, it's likely the PhD can write a poem in Shakespeare style. So that's what actually really this pre-training, we also call foundation model, also large language model is capable to do. So that's the moment we actually want to build up the RNA foundation model. So give a little bit more practically towards, the size of that is really machine learning is you need like a several hundred data set. Then, however, you need to give all the features towards what it is. For example, you need 100 like cat figure or 100 dog figure. Then you actually need to tell them what is the difference between dog and a cat. So you generated the prediction model. It's like you actually need to give a very specific features to the model to be able to learn it. So the deep learning is better, but you need a lot of labeled data, which means it will generate these features itself to do the prediction, but always it's label intensive. It's like you think about if we want to know 300 base, UTR, how it's corresponding to translation, then if individual you make a mutation, it's 4 to the 300 times experimental data you needed to generate. It's not feasible as actually most biological content. So what actually currently foundation model is really we train all the animal photos, then let it self-learning. So you give a little bit of data set, some cat like maybe less than 100, cat photo, dog photo. So you generate a cat dog classification model. You can do chicken, duck, everything. So basically you have a stage which you pre-training let self-learning model generator. Then you're adding the fine-tuning step, which then you can actually do individual task. That's why it's also called foundation model. So when we actually think about it, we start with our favorite plant transcriptome. We have these 1,000 plant transcriptome data set, which have a lot of diversity. So what we did is we put it into this kind of self-training machine to let it learn the feature of all the RNA language. So what we exactly trained is we take these 1,000 plant species, pretty much 25 million RNA sequence, 54 billion RNA nucleotide. Then we're using both RNA sequence, annotation information, also in silico RNA structure information as actually way as three things coming together that it self-train. So we ran on like a GPU, like a 7 GPU for like 2 months, generated this first plant RNA foundation model. So how we actually interpret that? So we basically do a fine-tuning step. So now we ask, okay, if I give you a 5 UTR of, let's see, Arabidopsis plant or rice, so all the 5 UTR, then corresponding translation efficiency, can you actually give me any pattern of that? So then we basically put our fine-tuning data set with Arabidopsis one data set, rice another data set, with actually their 5 UTR sequence around with their label of translation efficiency, and put it into this foundation model. Then we shuffling the label, then generate a random model, just actually as a contrast, so we know that's the background. So then we generate something called attention contrast matrix. So you subtract the learning from the truly data set, then subtract the random one, try to get a highlight which nucleotide is more important to its translation. So then we can average all the 20,000 genes 5 UTR in both Arabidopsis and the rice. What you can find a very interesting feature when you average these attention contrast score, you think about it, it's higher, that means it's more important. If it's lower, below zero, that means it's not important. So we can find a roughly 200 base close to ATG, these actually nucleotides tend to be important to the translation efficiency. More close, actually more important, no matter it's a sequence motif or structure motif. So then if you enrich this area, you can actually easily find the Kozak sequence. Just to let you know, I never tell the model there's something called a Kozak sequence. So it's actually found itself based on, this attention contrast matrix. So then we thought, okay, whether or not we can extract some structure motif. So we actually did a structure prediction, generate the seed library, then associate this seed library with our attention contrast matrix to overlap it. Then we did some enrichment to see what is a high translation associated motif, what is a low translation associated motif. So interesting, we can find these exactly every single gene you can find where this low translation associated motif is. So interestingly is we can find these 4 pairs of GC kind of stem loop coming up all the times. And very interesting is everyone knows when you do correlation study, you will see like a high GC corresponding to low translation efficiency. Now what we can do is we exactly know where this motif it is, where this gene have actually corresponding this motif. So you can really identify where it is. So we also interestingly found these kind of AGCU motif, which corresponding to the high translation efficiency. So rather than very strong stem, you actually have a relatively weak, but it's repeat like AGCU. So interesting in Drosophila, there's one individual case which they find in the 5 UTR, you have this repeat, you actually have a high translation. So now we can identify exactly what this motif it is, where they are located on which gene. So based on that, we can actually doing some manipulation. We're changing low translation to high translation. You can actually see reporter assay. We can validate that. You also can changing the high translation to the low translation. You can see we can suppress translation. So then, of course, another interesting thing is I just talk about the G-Codaplex. Everyone said G-Codaplex is supposed to suppress translation, right? So which G-Codaplex exactly in the transcriptome suppress translation, right? So in our actually foundation model, we can identify exactly 21 low translation associated RNA structure, RNA G-Codaplex really have a strong, significant impact on the translation. So this is one of the example. If you mutated this G-Codaplex into GA, so abortion A, G to A, then you will see this actually going higher translation. So then my postdoc said, okay, rather than we just do, one mutation changing, translation efficiency, can we turn in the gene as translation as what we want, right? So then we go back to this kind of attention, average contrast kind of trend. You can see close to the AUG, it's more important, no matter it's suppressed translation or, enhanced translation. So my postdoc did a simulation for predictor, gather this G quadruplex, move from the actually inside the close to AUG, then move outside to the 5-UTR. You see, based on the simulation, we can actually predict this importance getting weaker and weaker, nearly gone. So then we thought, okay, maybe we can actually see whether or not it's really work if, this is the case. So what we did is we take this native G quadruplex, which is suppressed translation, right? We actually move it close to AUG, you suppress more, even close to the ATG, you suppress completely. Then you actually move it out, move it out towards actually 5-UTR, end of 5-UTR. So you actually can see it's actually when you actually move out of nearly the end the beginning of 5-UTR, you see it's actually similar to your mutation. You completely abortion the translation suppression function. So we think, okay, this may be a good way, engineer the gene as to actually get the level you wanted rather than, just switch on or switch off. So again, we actually now invest more function towards not only translation, degradation, RNA binding protein, M6A. We think this foundation model can do a lot of things when you actually fine tuning with different downstream tasks. You can actually decoding what is exactly important for, function. And of course, so when you know these code, you can actually design the code. So we're kind of starting not only doing in the reporter assay, we're doing on the plant transgenic line, trying to see whether or not we can control, plant height is what we want, hopefully. So this is something still under progress. So finally, I want to acknowledge my group. So also all the funding agent, I needed to say we are more like a collaboration kind of lab. We collaborate with a lot of people. I really needed to mention is Ke Li, who is the AI scientist behind the whole foundation model. So he is located in Exeter. We actually collaborate a lot for doing a lot of interesting things. I think there's a lot of space on the interpretation of what a foundation model really learn. So we actually currently doing that. Recently, we actually got 8 million for this whole new scheme of AI for RNA biology. So if anyone interested to do a postdoc with us, please contact me. Thank you so much.
Thank you. Okay, so we're gonna have questions now. So people can either raise their hand and unmute or put it in the chat and then we'll collate and ask them. While people are coming up with the question, I actually have a question. I'm kind of intrigued by the relationship in vivo between temperature and salt conditions. Is it known whether the kind of ionic conditions in the cell is changing when the cell goes from 20 to 4C?
So currently so plant is a little bit more complicated because every single cell type in the root, for example, the plant root. So there's actually different cell type. Each cell type, the ion concentration is relatively different. So you have different level of potassium. So it's hard to see, okay, what exactly you're gonna actually really measure. So we do actually have some potassium treated and sodium treated is still on the way. So we think G quadruplex is likely to respond to majority, which we think like even molecular crowding, all these kind of conditions. So in the plant, the molecular crowding is more similar to the drought stress. So if you think about the drought, so that's actually induced kind of a similar things as molecular crowding. So we're actually heavily in kind of doing that. Interesting thing is currently based on our observation is there's a different set of G4 was induced. So it's not exactly always the code one is actually also come up in the drought stress or salt stress. So you might have a certain specificity.
I can also ask, I wanted to ask, like, there are different models, foundational models for RNA, and they don't are like group specific. So do they ignore plants, or like there's like so why your model is different? Like the source of information is different, and is the predictions that you did with other models would be similar, or there is something unique about the data set that you used?
So basically, is when we starting trying, it's really go back to 3-4 years ago when we starting these pre-training step. Of course, we actually very ambitious at the very beginning. We pretty much using the whole NCBI to do the training. But when we do the fine tuning, especially during the translation efficiency, which are really easy to see, right? So apparently, you actually always attention going to the SD sequence. It's really biased towards because 64% in the NCBI is actually bacteria genome. So that's actually causing quite a bias towards our learning. So that's why we currently separate, bacteria, animals, and also the plant. Maybe we needed to do, more specific species because imagine plant species are still a larger diversity there. So we think is really towards the question or fine tuning you want to do. Then your foundation model need to be on that. The more doesn't really mean better. So if we actually, our foundation model, when we do plant-specific downstream work, far more better than any, RNA foundation model when we do the performance test. It's really depends, what is really the things you want to learn. I don't think, it's really towards just one foundation model, you do everything. It's you might need to separate the system and really the question you want to ask.
Thank you.
Yeah. Also, I needed to say, because I'm interested in RNA structure, so my pre-training step, I put RNA structure there. But the majority actually foundation model, they don't put the RNA structure folding there. So I think we're actually doing more complicated pre-training at the data perspective. So that's quite different than almost all other kind of foundation model.
All right, we have a question in the chat from Tushar. For your SM structure-seq model, can you plug in an RNA sequence and predict the structural dynamics?
So basically is we currently is using the constraint. So if we see the mutation, we're using that chemical probing details to predict the conformation. So it's kind of relying on the chemical probing itself. So for just you can do control on just the RNA sequence, then see the dynamics. But the same as what we find is majority in silico prediction, the structures tended to be more stable as a thermodynamic more stable. But the in vivo one is tended to actually have a more single-stranded than majority in silico prediction. I don't know how much the in vivo can really kind of do the dynamic prediction, because you might need to validate afterwards with the more thermodynamic by physics method. Then the in vivo won't really help that much. If in vitro, of course, we can also do the in vitro single molecule one. I think that result will be more kind of close to the by physics analysis. Thank you.
I think I have a question. Yeah, so first of all, regarding the cold stress, where you could see like the formation of G complexes for sequences which are not extremely G rich, but they have like adenosines and probably T, I would believe. So is that because like during that stress there is A2I editing or something like that which is happening? I think I have worked on something related to that, like where during stress or salt concentration, A2I editing is like promoted, which actually in turn promotes the formation of G complexes. Did you observe something like that, like modifications?
We haven't actually aligned the modification data yet, but I think there's something. So plant majority M6A modification was kind of a lot. So we're actually currently working on Tranher's data on the M6A in the plant. So maybe actually there will be actually space towards see how much link with G-force formation. But the stress response mainly, so the temperature do actually have a more global effect even on the RNA secondary structure. So whether or not the temperature really actually induce a certain level of a modification, we think that might be, but you towards the really population, right? So the modification, how much proportion you really actually due to the editing causing the conformation forming equivalent, we don't really know. But that's something really interesting to see whether or not there's any correlation. Yeah.
Yeah, thank you very much. And I have another question regarding the training of like the pre-training that you were talking about. So you mentioned using secondary structure data. So I believe you utilized VNF fold for predicting those structures. Is it so? Okay. And did you also use like multiple sequence alignments? Maybe no.
Yeah, not at this stage because I
Okay.
give you an idea. So pre-training on the sequence level learning, it's already introduced a lot of conservation information crossing the plant species. So when sequence learn itself, the conservation somehow is already embedded into the learning. The structure part, I needed to say, because due to limitation of GPU, of course you can go more wide. So currently the pre-training we did is we had a 30 nucleotide window size folding of ensemble kind of RNA fold. So we're using ensemble rather than the lowest minimum free energy one structure. And we also did one with 100 base pair kind of window folding. So we didn't go beyond that because then you think about 1,000 plant transcript, how many folding structure CT5 it is. So currently it's quite limited, but we actually will input more afterwards. So currently it's only including 30 base and also 100 base kind of a window folding one structure there.
Okay, thank you very much.
I have a more systematic where you consider change in structure, perhaps for G4 difference between species, how different.
Yeah, we can read it to everyone. Are you planning in having a more systematic approach where you consider the changes in structure, perhaps for G4 differences between species and how the difference in the composition make them better adapted for growing in particular environment? Yeah, it's interesting.
Yeah, we actually do have a more systematic, but you couldn't really do 1,000 plant transcriptome for the whole G quadruplex profile. But we do have, apart from the rice, we have a wheat. We also now have a different kind of a brassica plant. You have a sequence interpretation. We want to see how much actually really influence and the genome-wide the content of that. We haven't do a lot of environment in different species yet, but we do have brassica plant, wheat plant, and all these kind of nucleotide diversity is more kind of a species to try to see exactly what we actually see. So that is slowly on different type of species, I needed to say. Yeah, I'm always available for question. If you actually guys think about you want to contact us later, just send us email. Our protocol also code is always available to almost everyone who want more information. So this is currently we want to promote and more people using our method or other type of thing. Hello, Eugene. Hi. Hi.
Hi.
Hello.
If I may, yeah. So thank you for this presentation. It's just like great. So two questions. So the VNA RNA predicted structures, they cannot predict G4 structures, right? So have you looked at the overlap of these G4 motifs, sequence motifs, and what structures VNA RNA predict for these motifs?
So currently the G4 we didn't really, so I needed to say is the reason we find in the attention matrix on the G4 is we align G4 motif with this G nucleotide repeat. And if attention come up with all these G repeat, as we predicted that likely to be G4, that's how we identify G4. So we didn't constrain fold anything with G4. So currently for the foundation model, we didn't input G4 because we think it may be biased the whole learning. So it's only secondary structure. For the G4 kind of a prediction on the VNF fold, so currently is so that's what we currently learning on the G4. So we also have a G4 prediction foundation model. So what actually happening is we think a franking sequence is actually influence G4 more than the G4 sequence. I mean, apart from the motif itself, like a loop comparably, all the franking have a lot of contribution towards G4. So we kind of validate whether or not that's the case. So we think it's actually can maybe better to help improve G4 prediction in the VNF folder. So that's we actually doing that now. But how much we can embed, help VNF folder to have a better prediction, we will see how the performance it is. But just based on our currently learning is actually the franking quite important, especially like a 10-10 nucleotide downstream upstream. So it can help later. Yeah.
Yeah, and pretty much the same question about the pseudonodes. So since VNRNA cannot do pseudonodes, I think like that's also a great bias toward just stem loops.
Yeah, so a pseudonode currently we couldn't really actually get it clearly to learn. So G4 is because population wise we can identify more, but pseudonode currently is we couldn't really have a better kind of a strong model to actually doing the pseudonode. Maybe actually individual rubber switch, we can actually doing some inverse learning, but that's really something we are testing. But I'm not sure currently we can do that. But the G4 prediction for the RNA G4, we actually may have a really better prediction. Yeah.
But just from my experience, IP-node can be pretty confident about the pseudonodes, and it's pretty fast to incorporate in large scale analysis.
Okay,
IP-node,
yeah, if we can discuss anytime. Yeah, and
no, it's not my tool. I'm just saying that from my experience, we have some tools that are pretty confident about pseudonodes.
Yeah, okay. Yeah, that would be great. I was saying that it's basically just using currently foundation model or any experimental data we currently have. This is the current stage we kind of we haven't really think about how to do the pseudonode. But if you're happy to using some foundation model or some our probing data, that would be great.